Hi, everyone. Thank you so much for coming today. I'm so excited to be able to be a part of this collaboration talking about forest threats, specifically hemlock woolly adelgid, which is what I'll focus mostly on today, and the way that science and art can work together to address those challenges. I'm Grace Haynes. I'm the extension aide at the New York State Hemlock Initiative, which is a part of Cornell's College of Agriculture and Life Sciences. So first, I want to start by kind of grounding us in what it is that we're trying to protect. This is a photo of one of our local forests, and I know it's kind of hard to remember what that looks like this time of year, but kind of remember what it's like to be outside in the forest, exploring. And one of the things I want you to notice about this photo is just how many different kinds of trees are represented here. So we have our hemlocks in the foreground. Can you see this? No, you can't. There's some hemlocks in the foreground. That big trunk off to the left is a more mature hemlock. And then if you look to the back and the rest of the photo, you might be able to see a lot of different species of broadleaf trees as well. Maybe you see some maple leaves, beech leaves, birch leaves, etc. And those are just Eastern North American forest tree diversity is just one of the things that makes it so special and so wonderful for us to explore. In New York State specifically, we have 18.6 million acres of forested land, making up 61% of New York's land area. And in those forests, there are over 100 tree species that are represented. And so that diversity is a really beautiful thing, that it's one of the things that makes our forests so magical to explore. It also can make our forests a little bit more vulnerable to invasive species. Just by virtue of there being so many different kinds of trees, that means that there's more potential hosts to insects that feed on different kinds of trees. And those invasive species in our forests, they've been impacting our forests for a really long time. As far back as 1869, spongy moth was introduced in Boston. That was its first introduction to North America. This is a moth that's not usually a direct killer of trees, but it does add additional stress to forests. And since its introduction, it's defoliated over 95 million acres. In the 1890s, beech bark disease was first detected in eastern North America. This is a disease complex, which means that there are multiple species interacting to harm the trees. And so this beech scale that you can see on the left side of the screen, that creates these openings in the bark that allow the neonectria fungus to colonize those trees, and that's ultimately what leads to their deaths. In the early 1900s, chestnut blight was introduced to the east coast of the United States. Chestnut trees used to be a ubiquitous piece of our eastern landscapes. Prior to the introduction of chestnut blight, they made up an enormous part of our forests. They were a huge tree and were familiar to most people who explore those forests, but now they're considered functionally extinct because of the chestnut blight. So that's one of the more extreme examples of an invasive species impacting our forests. In 1908, a longate hemlock scale was first detected in New York. This is a scale insect that lives on the needles of hemlock trees. It can also feed on some other species, but is primarily damaging to hemlocks. Then we saw the introduction of an additional disease complex, Dutch elm disease, in the 1930s. This is a fungus that's vectored by these two species of bark beetle. And this has killed over 40 million elm trees. In 1951, we first detected hemlock woolly adelgid in eastern North America, although genetic research has shown us that it was actually first introduced in the early 1900s. We just didn't find it until the 50s. And this insect has killed untold millions of hemlock trees. In 2002, emerald ash borer was first detected. This is considered the costliest forest pest ever to invade North America and has killed millions, tens of millions of ash trees. Beech leaf disease caused by this nematode on the left side of the screen was first detected in 2012, and this adds an additional stress to beeches, which are already harmed and threatened by beech bark disease. And then in 2014, we first saw spotted lanternfly in North America. This is another insect that's not typically a direct killer of trees, although in heavy feeding, it can kill grapevines and black walnut saplings. So this is just a subset of all of the different invasive insects that have been introduced to North American forests. And you can see many of these and the trees that they impact reflected upstairs. We have some insect and plant specimens as well as in the artwork. These are reflected as well. Yeah, so that gives us an opportunity to interact with them a little bit more directly as well. I'm hopeful that if you're here today listening to this talk, you already care about our environment, you already care about our forests, and you want to be informed on these issues. We're often really inundated with information on these difficult topics, but we don't always also have information about the positive actions that are being taken to address them. So I can't speak to all the research ongoing on all of these different invasive species, but what I can do is tell you about what work we're doing at the New York State Hemlock Initiative. And at the Hemlock Initiative, we work on hemlock woolly adelgid, that major threat to hemlock trees. So I'm going to take a little bit of time to share with you about hemlock trees specifically, what they do for our environments. Then I'll talk about hemlock woolly adelgid and the threat it poses. And then I'll talk about what we at the New York State Hemlock Initiative, or NYSHI, are doing to protect them. So let's start with hemlock trees. Hemlock trees are a really beautiful and important tree in our ecosystems. They are the third or fourth most common tree in New York State. And New York has more eastern hemlocks than any other state in the nation. And so this map here shows you the distribution of hemlock populations within New York State. And so you might be able to see from this map that they're most densely concentrated in the Adirondacks and the Catskills and the Tug Hill Plateau regions. So because hemlocks are so common, it can be really easy to take them for granted, but they are a really important species. They're slow growing and they live for a really long time. And so when we see these large, healthy hemlock stands, that represents a really mature forest ecosystem. That also means that it would take a long time for those ecosystems to recuperate after the loss of hemlocks. Hemlocks are a foundation species. That means that they define and structure their ecosystems. So because of their dense branches, they are cool during the summer, they're a little warmer during the winter, and so that means that they provide refuge not only for the wildlife that live in these forests, but also for us when we go and camp and hike in these areas. Hemlocks are able to grow in areas that many other things are not able to grow in. Looks like my formatting got a little bit shifted. Because they're able to grow on steep slopes and in shady areas, that means that they fill a niche in the environment that otherwise may not be able to be filled by other trees and plant species. They also are the base of the food web in their ecosystem. So besides providing shelter, they also provide a winter food source for a lot of different species. So these species are deer, moose, porcupines, as well as some of the smaller ones like tree arthropods, salamanders, and birds. They also often grow next to creeks and streams, and so they create unique habitats not just in the terrestrial environment, but also in the aquatic environments that they grow alongside. So because, again, of their densely branched twigs and branches, they provide a lot of shade, and that keeps the water very cool. They also can slow spring snowmelt, which keeps water cool through a different mechanism. And they provide additional ecosystem services as well. Because they have really densely branched root systems as well, they help keep soil in its place next to these riparian areas. That prevents not only the sedimentation of that soil, but also anything that soil may be carrying in it, like pesticides or fertilizers. They essentially act as a filter to keep our forests, our streams clean. And so all of those things are reasons why it'd be so devastating to lose hemlocks. This photo is from the Southern Appalachians, and And so this shows kind of what it looks like once HWA, once hemlock woolly adelgid has moved through an ecosystem. So in those areas in the southern region, the southern distribution of hemlock woolly adelgid, they're really doing everything that they can to save just the very few hemlocks that still remain. But in our region, we have the advantage of a little bit more time, and so we're still able to try and be proactive, to try and find hemlock woolly adelgid before it's reached this state, and to try and prevent this degree of hemlock loss, so that way our forests don't end up looking like that. This is what hemlock woolly adelgid looks like. This is if you're out in the woods and you're looking at a hemlock twig, you were to flip it over, this might be what you would see. So that's hemlock woolly adelgid, an invasive hemlock pest, and I'll be referring to it from here on out as HWA, which is its acronym for hemlock woolly adelgid. So again, you can see it here. This is what it would look like to you out in the woods. It looks like these white, waxy, woolly masses that you'll see at the base of hemlock twigs, at the base of hemlock needles along the twig. Here. So they secrete this woolly substance to protect them as a buffer from wind and cold, and that's also the place where they'll lay their eggs. If you were to zoom in, take all the wool off, this is what you would see. This is what a naked hemlock woolly adelgid looks like. And so that long, spinny, straw-like thing is called its stylet mouth parts. That's what they will insert into the twig at the base of needles, and they use that to extract resources from the tree. That's how they survive, and that's also the thing that damages tree tissue and ultimately can lead to the death of hemlocks. They can kill hemlock trees in 4 to 20 years, and that number is going to be closer to 4 if those hemlocks are experiencing other kinds of stress, things like drought, poor growing conditions, if the winters are too warm, or if there are other tree pests present like root rots. So this map shows you all the different species of hemlock trees worldwide. So to kind of orient you, we have Asia on the left side and North America on the right side, which is different from how we usually see them. And there are many different species of hemlock tree in these areas. The native range of HWA corresponds with these hemlocks in Asia and Western North America. And so in those areas, there's a shared evolutionary history between HWA and the hemlock trees. So that means that those hemlock trees have some natural defenses, and also that there have been natural predators that have co-evolved that are able to keep those populations in check. But here, where we are in Eastern North America, in HWA's introduced range, that's not true. They don't share that evolutionary history, and so that means our eastern hemlocks have essentially no natural resistance to HWA, and there are no native predators. So that means that HWA is free to spread unchecked through our hemlock trees and kill them, which is what it's been doing. If we zoom into New York State specifically, this map shows us the spread of HWA through townships. So those lighter colors correspond to earlier detections, and then the darker reds correspond to more recent detections. So in the 1980s, HWA entered New York in the Long Island and lower Hudson regions, and since then it's mainly been spreading north and west. This is a really interesting insect. It has 2 generations per year. So each of these generations is going to start with the eggs hatching. Then they'll have this period of time when they're a nymph that is mobile. We call that the crawler because it's the only period of their life when they're moving around. So they use that time to look around for a good place to stop and spend the rest of their lives. They settle on a twig at the base of a needle. And then they're going to go through 4 different molts until they reach adulthood, lay their eggs, and start over again. And so for the first generation, that's their overwintering generation. So in this first generation, they have a period called estivation. So they have a dormant period where they don't undergo any development, and that's why it takes so much longer. So they're going to be mostly dormant through the summer and part of the autumn in the overwintering generation. Their second generation is the spring generation, and in this generation they don't have that dormant period, so they pass through those 4 molts much more quickly, and it lasts only in the spring. So when they're these little crawlers, they look like this. They are super small, about 0.2 millimeters in length. And so if you see them without a microscope, they really look like just tiny little specks. And so because they're so small, they're easily able to be spread. They can spread on migrating birds. They can even spread on the wind. And if we are to go out into the woods at the time that they're in this stage, it's very easy for us to transport them if we were to go from an infested hemlock stand to an uninfested hemlock stand. And you might see them in this stage between April and June. After they find a good spot to spend the rest of their lives, they settle on twigs, and then they look more like this, again, obviously blown up. So this, for that overwintering generation, this is when they have that long dormant period through most of the summer. And so you might see them in this stage or not see them because they're hard to see from July to October. And at this point, they're very hard to detect because they're about the size of sesame seeds and they don't start producing wool yet until after their dormant period. So come the beginning of fall, mid-fall, they're going to begin to grow again. They come out of their dormant period. They start to feed and grow, and this is when they start putting on that wool. And so this is kind of when they'll look like that initial picture that I showed you, where there are these little cottony masses at the base of needles. And you can recognize them in this stage from about November to June. And then once they reach adulthood, they're going to start laying eggs. Then, of course, after the overwintering generation, those eggs will hatch and move through all these stages much more quickly. You might see them as adults or adults with eggs between April and May. So there are a few different reasons why HWA has been so devastating in eastern North America. First of all, they reproduce asexually, so that means they just clone themselves. All of the hemlock woolly adelgids that we see in eastern North America are females, so they don't need to find a mate in order to reproduce. And that means that just one single adelgid is able to start a new infestation of HWA. So that gives them this opportunity to reproduce much more easily without having to find a mate. They also have those 2 generations per year, which means that they have 2 different opportunities to repopulate if we are able to knock back their populations through some of the mechanisms that we use. And then, of course, they have no native predators in our region, which means that they're unchecked by the forces that act on them in their native range. So these are kind of things that we have to keep in mind as we are doing our HWA management, but we are doing HWA management. We have a couple of different ways that we approach this. In the short term, the only strategy that really works is using chemical treatment. Basically, if you want to save an individual hemlock tree, the only way to know for certain that you'll be able to save that is using chemical treatment. In the long term, we are releasing predators from their native ranges, and that process is called biocontrol, biological control. And so the idea here is that we'll be able to use our short-term management to preserve hemlocks, to preserve their genetic biodiversity and keep some of them on the landscape, while our predators that we're releasing, our biological control, is able to establish and spread on its own. So it's really these 2 things working together that will hopefully save our hemlocks. For our chemical treatment, our short term, we have 2 different products. We have imidacloprid. This is a very slow-acting chemical that can take up to a year to fully protect the canopy of the tree. But once it gets there, it lasts for quite a long time. So it lasts from about 3 to 7 years, which is an average of 5. And this one is widely available. At this point, until July of this year, it's legal for homeowners to purchase this on their own and use it to treat their own trees. And it's also fairly cheap compared to our other chemical that we use, which is called dinotefuran. Dinotefuran is quick acting. It can be taken up by the tree in as little as 3 weeks, but it doesn't last nearly as long once it gets there. So it only lasts about 1 year. It gives about 1 year of protection. And this is less available. Only qualified applicators are able to use dinotefuran, and it's also quite a bit more expensive. And either of these can be applied in different ways, either applied to the stem of the tree where it'll take it up through a spray along the bark or injection into the tree, or with a soil drench or soil injection. So that's our short-term management. In the long term, we're doing biological control. This is a long-term strategy that's also landscape scale, and we're still doing a lot of research on this. Basically what this looks like is we have two different kinds of biological controls that we use, two different kinds of predators that we use for biological control. We have a couple of species of flies and a couple of species of beetles. This is Laricobius nigrinus, one of our beetle species. This is from the Pacific Northwest, and it feeds on that winter generation of HWA. We've been releasing this since 2009, and we have really good evidence that they've been able to establish at over 30 sites in New York State. So we have a lot of good evidence that they're spreading, they're doing their work. This is what they look like when they're eating HWA. This is a hemlock twig, and all those little beetles are eating the HWA on it. This map shows you the places that we've released and recaptured Laricobius. So those pink areas, that represents where HWA is by township. Then those black dots show places where we've released Laricobius beetles. Then if you look at those gold stars, those are places where we released Laricobius and were able to recapture it. And the red plus signs show places where we've recaptured Laricobius, but we did not release it there. So those red plus signs show essentially what that represents is places where those beetles have been able to spread on their own without our help. So we have really, really good evidence that this insect is doing a great job spreading on its own on the landscape, establishing and doing that long-term control. But because these only feed on the winter generation, that means that the spring generation of HWA can be able to repopulate. And so that's really what we're looking at our Leucotraxis flies to help with. These are also from the Pacific Northwest, but these are a spring feeder. So they feed on both generations of HWA, and that means as they're feeding on that spring generation, they're theoretically complementing the efforts of our Laricobius beetles. And so we're hoping that with both of those together, we'll be able to knock back the populations, not just of the winter generation, but also of the spring generation. They eat HWA eggs, and we've been releasing them since 2017. And with these, we're still looking for evidence of them establishing on the landscape. So we're hoping to have good news for you on that front soon. This is what they look like. They're very tiny. If you saw them in the wild, you would have no idea the great work that they're doing for our hemlocks. But if you were to zoom in and look at them as larval, the larval stage, they're voracious feeders on those HWA eggs. So this map shows us, again, those pink spots are where HWA has been detected by township, and then the blue dots are places where we've released these flies. And this is supposed to say integrated pest management, which is the name of using multiple different strategies to try and control for a pest. So here we have our chemical control on the left side. In the center, that's a photo of our Leucotraxis flies, and then our Laricobius beetles are on the right. And so by using all of these together, we're hopeful that we may be able to protect our hemlocks. Now, that's all the science. Where does the art come in? Something that is often, an idea that's often shared is that science and art are these totally different things, or science and humanities are kind of these different silos. But really both of them are ways that we try and make sense of our surroundings. Both of them are very human things to do to explore the world around us and try and understand it. And so in that sense, they're not so different. And on top of that, art gives us this opportunity to understand and connect with things emotionally in a way that facts and figures cannot. And so this is just an example of a poster that we have hanging in our lab that's just kind of bringing to life the biological control and the way that it's fighting hemlock woolly adelgid. So I've shared some of the facts and figures with you guys today, but I'm really excited to know that after this, we're all going to be able to go upstairs together and experience this art and experience these issues through a more emotional lens, and really have that visual component as well. So yeah, I want to say thank you. If you want to learn more about us, you can visit us online or follow us on social media. You can always shoot us an email as well. And I'm excited to hear from Johannes next.
That was amazing and very interesting. So I'm standing now between you and actually enjoying the exhibition up there. I was asked to expound a little bit on the art-science interface, and that's what I hope to do. All right. I'm a soil scientist originally, so not from the humanities, but I started to become very interested in these shoulder-to-shoulder works with the arts and the humanities, I want to guide you maybe through a few motivations and a journey. Why does a soil scientist or scientist explore the arts? Why should it? And I think why must a scientist explore the arts? And vice versa, I would probably say as well. So for me, it started really with education, and some of you, Professor Anna Davidson, we asked also to be part of a course that I teach at the moment with Professor Verity Platt from Classics, and with Professor Kelly Presutti, who teaches in art history. We started about 10 years ago to invite only artists and performers, visual and performing artists, to our class on environment and sustainability, which is a required class in the major of the same name. And I think that opened up a whole new opportunity for students, but also for us to explore the ways artists think and approach areas. And Grace teed that up beautifully. One of the early visitors were Marin Wilson, an artist from Philadelphia, who worked with the 100 students that are in that class on visual observations of plants and soil and critters. We've been in the Schwartz Center with Theo Black this year, working on biophilia and how Hamlet is dealing with the environment. So all of these educational exposure for us as instructors, but also for the students, were really revelatory. An early one and really notable eye-opener was a talk by Bill Gaskins, an artist and faculty member in the Department of Art, who is also a photographer. He talked about his own professional progression as a emergent artist, and he did not use the word environment and sustainability once, but we were at the edge of our seats to figure out now how does that translate, this struggle for meaning, for creativity in the arts. And we started then talking more about that, about creativity in general, about chance, trial and error, unlearning what we think we know, and learning new things, and started also publishing papers about that. How different are artists and how they approach problems, how they turn around questions, and that was really interesting to see. We had wonderful artists come to us. This one was Karine Bonneval from around Paris, that worked with students on soils and plants, and how plants and soils interact. She was also one that very early on asked the quote-unquote stupid question, what sounds do soils make? And I asked, sounds, soils? I've never heard soils make any sounds. And what turned out of a stupid question is actually a pretty brilliant question. And so we teamed up with a lab of O people who work a lot with sounds, usually the stuff that flies around or capturing those sounds, but they have never stuck a microphone into the soil. So we found some people who were equally intrigued by these kind of stupid questions. And then Karine recorded sounds with so-called accelerometers that were in different soils around campus here, made soil sculptures from those soils, and then placed the recordings of the sound captured into these sculptures that the visitor can then experience the sounds of those soils. That triggered a whole project that then and then visualized that also as part of an exhibition. That triggered a whole slew of follow-up questions and initiatives, and ended up as a funded project by some entomologists here on campus that tried to use these insights for developing early warning techniques for pests in agricultural fields. And so there are some pathways, and that would not have happened without an artist asking a stupid question, which turned out to be really brilliant. Other encounters, now asking questions about the world of tomorrow and how we will be perceived. I'm doing a lot of work on biochar, and so that artist was interested in exploring what does pyrolysis do to not just biomass, but all kinds of other things, as you can see. Another question where I said, scratch my head, how will that work? And indeed, it was kind of interesting to experiment with that person, Nathaniel Stern, and figure out what else can we pyrolyze and what does it look like afterwards. And then he gave me license to pyrolyze anything I wanted, so I used Walden's Pond by Thoreau. And see what that does, and you can actually still read the book. It's just black, and becomes totally different color and chemistry, but is still the same morphology. You can still recognize it's a book. And I have the book that then came out of that also here, The World After Us, Imaging Techno-Aesthetic Futures. That turned around for me also as a scientist. The whole scientific process, and I brought some books here also about the scientific process by some eminent scientists as well that wrote about that, Rachel Garber-Cole came to the class, but also did a performance in the plant science building where she posed as an agitated audience member that grills this dinosaur about climate change and what does it feel like to get extinct due to climate change, because obviously the dinosaur must know all about it. The dinosaur never answers. So the piece is called 106 Questions to a Dinosaur, and the point is that it's not so much about the answer, it's really about what question should I be asking the expert. And I think that resonated deeply with me, which meant we started making question machines. So what about if this building and the plant science building doesn't have all these fancy machines that answer questions, but it has also machines that ask questions. So we started building question machines, and that's also what we do in classes now. Of course, that's very whimsical. Also exercise, what would be a question machine? What would it look like? What would it do? How can we refocus from rushing to answers? And also in education, why are we asking questions where the answer is already known to the instructor? Why are we not focusing more on asking questions where actually the question is so good that the instructor doesn't know the answer? I find that much more interesting. So we started thinking about what we pitched as dichotomy between novelty and utility, and definition. So how do we define things, and solution, and see, and we do this exercise with students in the sciences, where they would put designs and engineering, science, and art, and it eventually always comes out like this, that novelty is really perceived to be and can be used as an entry point for the arts thinking, whereas science and engineering is really focused on being useful and providing solutions, but not to come up with novel ideas and novel ways of working and thinking. And I think that this should give us pause in figuring out, now, what are we not doing right, and where's our thinking not up to snuff. The research process, really, when we start figuring this out, the scientists' view of the world and the artists' view of the world, they come out very often very differently, also in the scientific process. So starting to put things upside down a little bit and unlearn, doing things just very differently, starting to think about, so this was very, very stupid in a way by me. Can we grade by fire? And I took a lot of exams that were graded traditionally, and then we just burned them and recorded the burn time. And actually, there was a good correlation, as you can see on the lower left side, between burn time and the grade. So I think a lot of things work. We also started to make airplanes, paper airplanes, and recorded the flying time of the good grades and the bad grades, and it also correlates somewhat, as well as how long it will float in water. But I like the burning actually the best. It was a lot funnier and more fun. Yeah. We started thinking about, no, especially I observed that the power of observation that I think in education we start to go back, and we're not really focusing on that with a project with the artist Ella Ziegler, and where we looked at the pivotal research by an eminent scientist called Barbara McClintock. Some of you might know her. Historically, she received all her degrees here at Cornell, but then couldn't get a job here, had to move on, and eventually received the Nobel Prize in Physiology or Medicine, 1983, and for a piece of research that is called the breakage fusion bridge cycle, or jumping genes in plain English, how inheritance works, and she did that on maize. And so the point in all these interviews with people who knew her and know her research was that she could, through looking at the chromosomes and looking at the different kernel sizes and kernel shapes and kernel colors of her Indian corn that she, by the way, grew somewhere down there. There's also still a Barbara McClintock shed down there that you can visit. Well, not really, you can go walk around, which would be great if that was eventually maybe a little museum or a commemoration of this, probably one of most important scientists that we ever had here on campus, and nobody knows her. But when she looked at a corn cob, she saw jumping genes, whereas I usually see food. And I think this lack of perception and emphasis on this observing and thinking that we also don't. We use now a lot of visual recognition software. All the students usually use statistics to look at data. They don't use their eyes anymore. I usually tell my students, don't give me any statistics. Just give me a graph and just tell me what you see in your own words. And if there's something that we should follow up on, then we can use mathematics. But let's first just look and see whether there's a story in these data. We started a broader conversation with across campus in the humanities and the science about an interdisciplinary center about 7 years ago, and came up with some guiding principles that came out of that. With different, that was COVID, that was all online, then in 2020, and honed in that actually the what are we working on was far less important to participants of these workshops than who was in the room, and especially how did we work. So the how emerged as a really important part of the conversation. How do we want to work? And so the practice itself, and what works for people across the disciplines, and the word discipline, of course, is problematic in itself. To discipline means a lot of different things. So we came up with maybe a little bit tacky line of undisciplining the university through shared purpose, practice, and place, making the point that the highest impact for undisciplining you receive when you think more about how do we want to work. Well, what is the how rather than the what? Whether I'm working on electric cars or soils or on unpronounceable pests, that doesn't unify me. Yeah? I don't know anything about electric cars, but I can have a very good conversation with somebody from the other side of campus about how I want to work, but not about what. So the what doesn't really unify people across different disciplines, very logically, but that came out really very, very clearly. And that was the impetus to know where I was going, that we needed to start a place that was profoundly undisciplined. And we call it the soil factory for obvious reasons, that it was a soil factory before we got there, and started to bring people in from all walks of the disciplines. So deliberately crossing the boundary of the institution in order to conduct a truly undisciplinary experiment. And so some defining features are, if you ask 5 people what it is, then you get at least 10 answers. And that's exactly the point, that it is something different to every person. That you really, the agency is with you. If you want something to done, you have to do it yourself. And that the precarity and temporality, who knows what the future brings, especially in these times. But it fosters, it gives you a license to explore. And maybe this is also a point about these times, that we need to take that license. Another principle of the detritus to figure out what are things that we want to keep from our activities, and how do they influence our future activities and thinking. These are just some impressions. There's a couple final slides of what people have done there, whether in performing arts, in music, in collecting indigenous herbal medicines, ceramic studios, dark room, and I brought a publication from the soil factory that talks about invasive species and makes the bridge between the invasives of a species and the foreigner, the humans that coming in, and how they correspond and what we can learn from each other. And a pertinent question that I'm asking also now for the university community, and would like to follow up more with that, is that need for us to, as a community of practice, to become a community of purpose, and whether the community is really building community is the precondition for productive science, and how art is catalyzing that. And another question, whether now science and art merging is the basis for community. And you can see that little guy is experimenting there very heavily with circular economy. Food and bringing community together is, I think, essential, and that's why I'm excited that you are here now, and that we get maybe some nibbles and some conversations at the next stage, and having that human connection. And these social practices are art forms in itself. So Soil Factory is a sandpit, and it had to be outside the university. We tried to build something like that inside the university and failed. So you have to, I think, and that's maybe a lesson learned in itself, that you have to get outside the university to build this kind of communities across disciplines. Exciting aspects of using different wastes for making different materials that also again came from an artist and not from a scientist. So I think ideation is a group activity, and welcome you there, but also very excited to be now part of the Exploring with You, the exhibition upstairs. Thank you very much.
I think we have some time for questions for Johannes and Grace, and so should we start with that?
So you said that there were only 30. You said that there were only 30 different places where one of the species had been released. It seemed like there were even fewer for the other. What's the future looking like? I mean, can you go faster? Can you release more? What's going on?
Yeah, so basically the focus of a lot of our research right now is figuring out what it is that makes it work better, and figuring out where it's established and kind of what we can do. So that is really the question, is how can we make it go faster, et cetera. We don't necessarily have an answer now, but that's kind of the focal point of a lot of our research, is how can we get that to move more quickly and be more effective.
your biocontrol agents, the fly and the beetle, they're raised in a lab, correct? Is there limitations on your release time and release factors and, you know, the pace at which you're going because of the production of those? And are they produced in a lab in the Northeast here? And if they're being produced in a lab, I still don't understand how being produced in a lab and then they are successful out in the wild. So could you touch on those things?
Absolutely, yeah. So basically, what the process looks like, they're kind of produced in a lab, but essentially our director goes out to the Pacific Northwest where they're native to, collects foliage, and then ships that back where we have a very carefully controlled quarantine facility. And so then we take that foliage. We have a few people in the room today who actually do this process specifically. We take that foliage, we put it in these, within our quarantine facility, within these cages, and then they extract the species, the biocontrol species, from that cage. And then we package them up. Basically, by keeping them in the quarantine facility and collecting them really cautiously, we avoid the risk of the Western type of HWA being introduced here. So it's basically just a safer way for us to collect that. And yeah, does that answer, did I address your? Okay.
So how long, I mean, when you're talking about, okay, Pacific Northwest, and then here, and then releasing,
Right.
how long does that take? I'm sorry.
Right, the question was, what's the process, what's the timeline?
Yeah.
Taking them from the Pacific Northwest to here to rearing them. So that, it does depend on basically what their life cycle is anyway. And so that means every year our director goes out, he collects foliage in kind of promising locations. And so what the constraints are on the number of species and the timing is they have to be collected at this time of year in the winter to early spring, I guess late winter to early spring. And then we get varying degrees of production of those species used for biocontrol. So that there's a number of environmental factors probably impacting them in the Pacific Northwest that we don't necessarily know in a given year. I will say though that looking at our numbers, I believe our production has increased every single year, and you can see that in our annual report. So I am still relatively new to the organization. I'm not sure exactly whether that is because of the process of selecting those sites or the process of selecting which twigs to clip and ship. But we do know that our numbers are getting better each year. And we also we collect data on how many of them survive in our quarantine facility, and those numbers, the percentage that survive and the percentage that are able to be released, that's also improving year by year. So there are constraints, but we're doing our best to work with them and to learn from them so that we can do better each year and hopefully release more and get them to establish more quickly.
Hi, thank you so much for speaking with us. So a lot of the times when we introduce biological controls into an environment, they kind of end up creating more problems later on. So what was the selection process like? Like, how did you select the beetle and the fly?
Yeah, so I tend to think that we have learned hopefully a lot from our past mistakes. And so what the process essentially looks like usually is putting them in some kind of a trial to see if given the opportunity to feed on and lay eggs in and among other species besides their target species, do they do that, and do they survive if they do? And so that is the rigorous process that they have to go through before they're approved by the government for us to release them, and that was done for both the flies and the beetles before they were released here. Yeah.
So I guess related to that, for the beetles and flies, are the adelgids the only prey, the only thing that they eat? And what would possibly prey on the beetles and the flies?
They are yes. For the first part of your question, yes. The only thing that they eat is the hemlock woolly adelgid. And there are a number of parasitoids that our lab has done a little bit of research on in the past. I would have to learn more about that to be able to give you a more thorough answer. I'm not certain, but I know that there are a number of parasitoids which can impact our biocontrol species. Yeah.
Thank you. With your chemical control of the adelgids, since they are asexually reproducing, are you concerned with them mutating to have resistance to that and then being asexually reproducing that and then that being a concern? Or are there breeding populations of adelgids that have some resistance? Thank you.
Yeah, so there are no breeding populations, period, in the United States. I think that the question about whether they can develop resistance, the asexual reproduction kind of cuts both ways. So it means that yes, if that resistance was able to develop somehow, it could spread more quickly, but it's also a lot less likely to develop because they're cloning themselves each time. And so the amount of genetic variability in the populations is super, super low. They're all essentially like the same individual. And so it's the fact of them reproducing asexually takes back a lot of that risk actually in some ways.
I had a question for Johannes. Yeah, I'm actually the artist showing upstairs, so nice to meet you. I actually had a question for you. I love the graphs that you had about the different outcomes for artists. I wonder if there's a ratio between fun and useful outcomes. I mean, maybe you'd like to comment on that.
That's a great suggestion. Yeah, I will follow up on that. Yeah, so I think a big motivation for, and this is sort of the emotional side that also Grace mentioned earlier, and that resonated with her, is very important for me, and I would like to explore that more. Yeah, so because I think we always think about science as hard and facts, right? A plus B, C, or something like that. So it's very clear. There are no emotions in there. There's definitely no fun in there. But I mean, but I think I think nothing can be further from the truth. And maybe again, Barbara McClintock, now in her memoirs and in her interviews, actually we did a button which I didn't bring, but when Ella and I went to the Women's Hall of Fame up in upstate New York here, where she was also inducted to, and looked at the archives and the transcripts of the different interviews that she gave, especially around when she received the Nobel Prize. I was just reading it and gave it to Ella, and Ella immediately zeroed in and said, "Oh, look at it." In the transcript, every time she was asked a question, there was the word laughter in brackets in the answer. So obviously, the most important thing of the answer was she found this all hilarious. And I think she connected emotionally also with the plant. She also said, I did not deserve the Nobel Prize. The plants showed me the way. So I followed their lead. So I think the emotional responses and fun is a very big guiding moment also in the sciences, and not something that we learn right about at all. Yeah, no, thank you for that. Yeah, love to talk more about it. Thank you.
Yeah, thank you so much, Johannes and Grace, for this inspirational talk. And before we're wrapping up and heading up to the exhibit, I'd like to thank my colleagues, Sean Taylor, Evelyn Frati, and also Jenny Leonaford, who is not here today, but she's with us in spirit. And we will go up there very soon, but let's thank Grace and Johannes again for their wonderful talk.